Dr. Ekaterina Gribkova is a postdoctoral research associate affiliated with the Coordinated Science Laboratory and Neuroscience program at the University of Illinois at Urbana-Champaign. She works with Professor Ron Gillette, who's with us today, primarily focusing on computational neuroscience and neurobehavioral studies using marine invertebrates. She received her PhD in neuroscience and her BS in mathematics and molecular and cellular biology, both at the University of Illinois. Katya, so glad that you could make it today and join us today. You have until the top of the next hour. We're really looking forward to hearing your presentation. So please go ahead and share your screen and take it away.
Thank you so much for that introduction, and to everyone for joining me here. So today I'll be talking about the evolution of memory and how we're going to go from simple associative learning to something as complex as cognitive mapping. So memory is one of the most important elements that can make an organism's behavior more adaptive and complex. In nature, we can see the increasing complexity of memory in examples like these. Some of the simplest organisms, like single cells, are capable of non-associative learning. And so E. coli bacteria, for instance, do random walks in search of food, and they can adapt to a homogeneous chemical environment through habituation, which lets them increase the randomness of the movement. So fairly simple adaptation. So classical conditioning is a bit more complex, but it is the simplest form of associative learning. And this is where a neutral stimulus is paired with a biologically potent stimulus, such as food, reward, or even pain. And so that the organism learns to respond to that neutral stimulus, essentially. And so there are some studies that suggest classical conditioning mechanisms in starfish and sea anemones, particularly when like light or darkness are paired with a food reward or painful shock. Though there's still a lot of debate over this, so nobody really knows. Higher order conditioning, so quite a bit more complex, is where neutral stimuli, multiple neutral stimuli can be associated. And this can be found in gastropods, such as the sea slug Aplysia californica, so a common model organism. There's no clear evidence of spatial learning in gastropods, but there is in cephalopods and other invertebrates, such as honeybees and jumping spiders even. Octopuses and cuttlefish in particular seem to display homing behaviors, learning of sequences of stimuli, observational learning, and likely even simple episodic memory. So at the furthest end, we see some of the most complex of memory processes, like cognitive mapping, which we can think of as the mapping of objects, concepts, and the relations for goal-directed use. While we can certainly see these processes in us and primates, there are some less common examples, such as baobabs, which actually have quite a well-developed aesthetic sense. And so how do we go from, say, mechanisms for classical conditioning to those of sequence memorization and cognitive mapping? Where can we even start? For us, a particularly interesting and accessible organism in this case is a special gastropod called Probranchia californica. And so here in this video from Patrick Webster, the predatory sea slug, Probranchia, is actually hunting for a small camouflage slug. So see if we can spot it maybe. And it's tracking it only as by its odor plume. And so here it actually hones in on its prey. And it's going to make contact in about a few seconds. So you see it actually try to escape, but it doesn't make it. So it's now consumed its prey. And so Probranchia is a generalist forager. It consumes a wide variety of prey. So it's quite important. It can actually learn the odors of specific prey and associate them with positive or negative rewards. So in this video, Probranchia is introduced for the first time to a colorful little slug called Flabolina, or the Spanish shawl. So our predatory sea slug actually smells the Flabolina, approaches it, and is about to find out it's actually not very tasty, as it receives a mouthful of stingers. So you see that it does that ejection response. So it ejects the prey, and eventually it does an avoidance turn. So it starts kind of turning away from it. And see, it just starts kind of slugging away in the opposite direction. So meanwhile, we see the Flavolina doing this desperate little escape swim, which isn't particularly effective because there isn't any current in this lab setup, basically. So about half an hour later, so let me just kind of fast forward through this. Probranchia and Flavolina are brought together again, and we see here as soon as Probranchia senses the Flavolina's odor, it immediately starts an avoidance turn, and once again just slugs away in the opposite direction. So we see that this is an example of simple associative and reward learning, as the Probranchia associates the Flavolina odor with that painful experience it had, and later on avoids it. So using the circuiting behavior of this animal, our lab modeled the core approach avoidance decision in foraging in a simulated cybersug. So here, sensory input, so this is a fairly simple diagram, but it's the sensory inputs come in to a number of different elements, and here incentive determines the overall expected reward, essentially, for the sensory inputs coming in, based on the learned associations and motivation, like hunger. While somatic map here was coming in, this describes a place code for the average site of sensory input to the sensors, which basically allows the forager to estimate which direction the strongest stimulus is coming from. So it's really a template for making kind of motor turns towards odor sources. And so here's that same network again with a few more details. And the core of this model is the integration of several different elements in the feeding network. And so this determines the final approach avoidance decision, really the motor output that approach avoidance turn. So satiation in this case is basically playing the important role of suppressing feeding behavior. It's kind of like a negative feedback loop. And the majority of this circuit, just note that it's basically almost hardwired in the sense that the only part where learning plays a role is setting up the expected reward for incentive, which does not solely determine the approach of Winston, but really it biases it, right? So it biases it through this through the feeding network. So the calculations in this network are actually quite simple, as it largely uses sigmoidal relations. And if anyone would like to see more of these, I have some slides at the end, so let me know. So the Cybersex simulation is shown here. Right, so let's zoom in on that. And it's a virtual slug encountering different kinds of prey. So learning their odors. And the red dots here are noxious prey, so let's say the Flavolina. And the green are just a beneficial prey, right? So not painful to eat. Let's call them Hermacinda, which is another type of low slug that Probranchia can encounter. And so over time, we'll see that it learns the associations for different prey odors, and eventually it will result in a greater preference for Hermacinda, right, the beneficial prey, over Flavolina. And so as we go through here, right, so it hasn't gone very many of the Hermacinda yet, but as it does, it actually forms a preference for it, right? And so this is not the only thing. So learning is not really the only thing that affects this behavior, right? So when it's very satiated, it's actually going to avoid all prey. It doesn't really matter what kind they are, even if it's the beneficial kind. And when it's very, very hungry, right, so it's going to actually go for all prey as well, because it's desperate. And so that's just like the real Probranchia. Now, this cyberslug has very simple learning goals for forming associations between prey odor and reward. And this is the Oskolo-Wagner learning goal, which models classical conditioning, and it's considered to be among the simplest of reinforcement learning algorithms. So basically, V here is the predictive value of, let's say, an odor stimulus I. And in its simplest form, this rule updates the associative strength between that specific odor and the reward each time the forager eats, right? So every time it kind of collides with that, with a prey item. And so this update is based on the current value, right? The odor salience alpha, the running rate beta, and the maximum associative strength. So note, this form is just a simple pairwise association between an odor and the reward. So with Cybersex, we had a simple yet fundamental framework for decision making, right, upon which we could build. In a sense, Cybersex is really quite expandable, and as we'll soon show, even the addition of like just a few more lines of code for a simple non-associative learning mechanism can make its behavior more dynamic and complex. So we first set out to give our Cybersex a more realistic affective valuation, or an ability to evaluate stimuli based on not just the stimulus qualities that it might learn, but also based on Cybersex's own experience and internal states, really. So we expanded it to a model we now call ASIMOV, as an homage to Isaac Asimov, the sci-fi author who was among the first to introduce the ethics of robotics. And so for this, we added a simple homeostatic reward circuit. So here it is. And with this addition, we realized that we actually had the emergence of addiction in the presence of high reward stimuli. So here's the Asimov simulation now, with the yellow dots representing high reward items. And these are simply called drug, basically. And so these provide no nutritional value. So only high reward. And we see that over time, the forger forms a very strong preference and addiction, really, for the drug items. So as we go on through here, we'll see that the drug consumption goes up pretty high. And so how is this done, really? This is really the structure, the very simple structure of Asimov's homeostatic reward circuit that we added, or the HRC. So it receives a reward input from prey and drug consumption and outputs a reward experience to modulate appetite state. So it's a bit like a negative feedback loop that diminishes with frequent input due to homeostatic plasticity. So reward experience can be considered as a dynamic hedonic tone, almost like the mood of the forager, so to speak. So by the equations, we'll see that due to the homeostatic plasticity mechanism in the synaptic weight, W, right? So there's that synaptic weight. We see that it's effectively, so the reward experience is effectively monitoring the change in reward input. So when there's a reward input, let's say, right? We see that a reward experience initially spikes, but then goes back to baseline even with ongoing reward input. That initial spike kind of satisfies the forger's drive for reward, right? So it's search for reward, reducing the appetite state, right? But here it's now essentially desensitizing or getting used to that reward input because of that homeostatic plasticity. And so when the reward input ceases, the reward experience plunges into the negatives because of that sudden change, just as you might expect in withdrawal processes. So in severe addictions, right, withdrawal is a very miserable process. And so over time, again, right, as it goes on, it goes back to baseline. The reward experience goes back to baseline, in effect, resensitizing. However, note that even with that recovery from withdrawal, even though it kind of gets back to a normal reward experience, this strong learned association, right? So because of that associative learning that we have for the higher reward item, especially, it's still present, and so it's giving rise to cravings, essentially, right? So even though that recovery happens, the cravings are still there. And so here's a simplified summary figure of the behaviors that the agent exhibits in addiction and foraging cycle. And so if the forager starts out satiated and not in withdrawal, let's say, and is exposed to drug, that learns that association and begins high rates of drug consumption. Right? So it forms that association and basically goes on a drug spree. And so basically, because the high reward experience, that initial high reward experience suppresses the feeding network, it ends up essentially ignoring hunger in some sense. And so it ends up quite miserable, malnourished and intoxicated, essentially. So over time, that desensitization worsens, so that initial spike drops off. And if there's not enough drug to sustain the state, then the forager actually ends up quite miserable, hungry, and in withdrawal. And this decreases prey selectivity. And so it actually leads to a different foraging pattern where it overconsumes prey. And so it becomes kind of satiated in withdrawal during that withdrawal state. And so over time, it will recover from withdrawal. So it will become kind of satiated and in withdrawal. But with cravings, this still continues that kind of vicious cycle because that association is still there. And so for a more dynamic view of this cycle, we looked at the forager's behavior in an environment with changing availability and reward quality of drug in different phases. So these are the plots of the numbers of prey or drug consumed. And in this first phase, with no drug, the forager develops a preference for the beneficial prey, as we might expect, as we seen before. And so once the drug is introduced and the forager tries it, we can see that the preference for drug really goes up. Right, so there's a pretty high consumption of it. And so initially it goes pretty high up, and the prey consumption goes down a little bit during this time. And so this is because the high reward experience essentially suppresses the appetitive state, right? So it increases selectivity for the drug, essentially. And so we see these mini withdrawals happening as time goes on, as it desensitizes to that reward input from the drug consumption. It actually desensitizes, and these mini withdrawals are basically this oscillation of reward experience between negative and positive values. So you see like very sharp kind of changes in foraging patterns here. And then once we actually start a full withdrawal, when drug is completely taken away, then it just plunges, right? So the reward experiences plunges into the negatives, and well, the Cybersex or ASIMOV forager becomes fairly miserable. And during this time, we have an overconsumption of prey. So because it's gone into the negatives, it's essentially exacerbating hunger effects and driving that consumption, that kind of decreased selectivity and overconsumption of prey. So as it recovers from withdrawal, it kind of goes back to normal, right? Preferring the beneficial prey, but we still have that craving, right? So that high associative strength for the high reward input. And so the way we, like pretty simple, oversimplified way we remove this a bit is we introduce a drug without reward. So basically the same context, same odor, but the drug item, that item no longer has that very high reward input. And so using the very simple Ascoli-Wagner learning rule, as if we set lambda to 0, essentially we extinguish that association, and it kind of goes back to the normal foraging rates. Again, it's a bit simplified, but we show that with ASIMOV, using that original Cybersex circuitry for foraging decision and adding this simple homeostatic reward circuit, we can get more complex and dynamic behavior, which includes those hallmark processes of addiction and altered foraging patterns. And so this demonstrates the expandability of Cybersex, or now ASIMOV's core circuitry. But the Cybersex and ASIMOV that we've shown at this point have very simple associative learning, where the forager forms the most basic pairwise associations between neutral order similar rewards, or really the level of classical conditioning. So how can we go from these simple, let's go Wagner learning goals and develop ASIMOV further for higher order conditioning, which has been shown in gastropods? So just as a preview, here's what a higher order conditioning paradigm might look like. So here is a naive forager that's fixed in place, and it'll be presented with different odor stimuli, which it can turn towards to or avoid. So let's let this go. So first, the yellow odor is presented, followed closely by a wood input. And we see that there's no appetite turn towards that yellow odor because it's novel, right? It's only just formed association for it. And so now here we see a pink odor followed by yellow, and it turns towards the yellow. And so because that's as previously learned. And lastly, with the pink being presented, and now it turns towards the pink, right? So with this sequence presentation, essentially this is demonstrating higher order conditioning. So we see that last approach, for instance, because the forager has associated the unconditioned stimulus, the pink odor, previously unconditioned, with the yellow conditioned stimulus that were before. So essentially, in higher order conditioning, we can form pairwise associations between neutral stimuli as well, not just between a neutral stimulus and a reward. And so note that if we can also encode the relative order or timing between stimuli, in addition to this naturally extends to learning of sequences of stimuli, which gives us the beginnings of simple episodic memory. So episodic memory, as you might know, encodes past experiences with specific contexts and allows us to learn and recall sequences of events. So now from an evolutionary standpoint, we have a fairly good idea of what would have shaped and supported the emergence of episodic memory, which would include the behaviors like establishing territories, mapping them, home and niche construction, as well as navigating between home and foraging sites. However, the circuitry behind the emergence of episodic memory is not well understood, particularly if we're looking at invertebrates. We do know, though, at least in mammals, that the formation of episodic memory in cognitive maps is mediated by the hippocampus. So this is a specialized brain structure which contains a variety of network architectures and plasticity mechanisms. Hippocampal place cells, for instance, are certain neurons that are associated with specific locations or contexts. As one follows a path, like a mouse running a maze, for instance, a sequence of active prey cells is formed, so it can be learned, and it can be replayed in memory. Backwards replay, quite interesting, are actually specifically associated with processing and encoding of rewards. So in the hippocampus, we have this auto-associative network, the CAII, which receives sensory and contextual input for basically place cell sequence formation. And sequence salience, right, so kind of the values of those sequences are assigned in CA1 by, so it's thought to be by pairing active prey cell sequences with reward input, essentially. So now many models of the hippocampus are highly detailed and require significant computing power. So we asked really what is the simplest hippocampal-like learning goals for sequence memorization. To this end, we created a simple model of episodic memory, the feature association matrix, or FAM, that uses some of the simplest associations, associative learning goals, and architectures. Essentially, the FAM chains together pairwise associations to memorize a sequence and assigns reward values to it. So we look at each pairwise combination of inputs. We basically have a set of matrices where each element is like a synapse that learns the strength of the association, meaning how much overlap there's between two inputs, the order or which input comes first, and the expected reward, meaning how close in the sequence are these inputs to a reward. So here's an example with a little slug agent encountering the sequence of orders 1, 2, 3, then reward. And in the so if I let this go, in the first two strength and order matrices, you see almost light up as associations are formed and chained together to basically make that sequence. And once the award comes in, so right there, let's see, there we go. So that backwards replay, basically this is where the expected award values are assigned along that same chain that was formed. And so this is how salience is assigned to that sequence. So now there won't really be enough time to look at these equations in detail, I think. So please let me know if you'd like to take a closer look at these. But here are the specific calculations of eligibility traces for each sensory input, so like short-term memory traces that last longer than the inputs themselves, as well as the strength and order and expected award value calculations for them. So, and this is like, let's say between a pair of inputs, I and J, for instance. The strength calculations are, I guess, a bit more related to symmetric Hebbian learning goals, while the order calculations are more likely related to temporally asymmetric Hebbian rules. So when the award input comes in, the expected award values are assigned using those previously established strength and order values. And so now the forger, with all this, the forger can calculate incentive across all sensory inputs, which can let it know basically which sensory input to approach or move away from in order to traverse a learned sequence and reach the endpoint to obtain an award. So for a simple demonstration, similar to the one we've shown before, here's ASIMOV's forger with the FAM learning temporal sequences. So I must note that we have fixed the satiation and the award experience at constant levels, so we could get a better sense of how the FAM specifically affects foraging behavior. So we present this temporal sequence of odors, of which the last only is given a award, so 1, 2, 3, then award. And the inputs here don't overlap, but the eligibility traces do. So those short-term memory traces do. And so we can see the actual simulation. And note that for that first presentation of the sequence, the forger does not turn towards the odor sources because it hasn't encountered that award yet, essentially. But once it does encounter the award, it reassigns values to those odors. And now on the second presentation of the sequence, it actually turns towards these odor sources. And this is because it's assigned value to them. And you can notice this by the incentive values that have increased from that last presentation. And so here's the same kind of higher order conditioning paradigm as we've shown before. And in this case, so it's a little bit more complicated than that sequence before. But in this case, this is a pairwise presentation of odors, a bit out of sequence in some way. So odor 3 is paired with award, odor 1 is paired with odor 3, and odor 2 is paired with odor 1. So in each case, the forger only assigns the value to the odor and turns towards it once it has formed the associations. And experienced that award, right? And so we see that it's basically chaining them together. So the award values here are interestingly, they do fade over time because the yellow odor source is essentially being presented multiple times without the award anymore. So it kind of shows that the forger can forget and extinguish any associations that are no longer awarded. And so again, this is just shows that higher order conditioning paradigm again. And now here's a forger, so same mechanisms. Now we can see how it learns a spatial sequence with the FAM. In this first run, what will basically happen is it will encounter 3 odor sources, of which only the last one, right? So only the yellow one is awarded. And in all the subsequent runs, we see that the forger always goes for the yellow awarded odor source. And it it does this often navigating along the memorized sequence and using the other odor sources as landmarks. In some cases, it can, if we look at a bit further on, it can even go through this odor source in reverse direction and just come around again. I think it was somewhere here. Oh, let's see. There we go. So it basically just uses these this sequence to find that yellow odor source. And so we may notice an interesting problem here. What happens when the odor sources are spread far apart and don't overlap, unlike these? So in that case, sequence memorization will actually fail for spatial navigation because there's no way for the forger to travel the gaps. And so for some additional inspiration in this problem, we can look at a sister taxon to gastropod mollusks, the cephalopods, and in particular, the octopus, which almost resembles a more complex and overdeveloped sea slug. So the octopus is quite adept at spatial navigation. Here we actually have one in our lab, emerging from its den and eventually kind of exploring. So from many different experiments, both in the lab and in the wild, we know that octopuses, in addition to higher order conditioning, show learning of sequential tasks, effective spatial exploration and homing behavior, path integration, and even observational learning. Furthermore, the octopus has neural architecture in its brain that is quite similar to that of mammalian hippocampus. It actually has also a very effective vestibular system that can communicate head direction information. And so that last one is actually kind of a key point. So from our previous simulations, again, we note that sequence memorization allows learning in traversal of overlapping unique landmarks. It does not, however, allow the agent to travel the gaps when landmarks are far apart. So we need something more. We need to also encode the distance and direction between the landmarks. Therefore, the addition of path integration allows learning and traversal of a map of non-overlapping unique landmarks. And so this may be the basis for cognitive mapping. So we can construct a spatial map through the following steps. After each landmark, path integration can provide an estimated displacement vector, which is essentially is obtained by a vector summation of scaled headings along the path. So this displacement vector, or the distance and direction between any two visitor landmarks, is memorized in the FAM. So it's kind of like a memory vector. And the forger can then use the memorized vector as a guide for making shortcuts between landmarks and even correcting itself if it takes a detour or veers off course. And then this is just through simple vector subtraction. Note that there is evidence of similar calculations in the mammalian hippocampal formation, particularly with good cells supporting vector calculations and vector trace cells forming a code for vector memory, which might even support abstract cognitive mapping. So here's ASIMOV, now transformed into a little cyber octopus with this simple path integration system, which relies on head direction and distance traveled, which is that slitherometer there. And essentially, this allows it to memorize the shortest distance and direction between any two odor sources. And so we'll see after it encounters all three odor sources once, it can, so if we just wait a bit through here, it can then use the map it has memorized to actually make shortcuts. And we'll see that coming up at this point. And so here are all the, if we fast forward a little bit, jump a bit in time, these are all the paths it's traveled. And essentially after that first encounter, it's making all of these different shortcuts. And so there's kind of an interesting little case where it overshoots this landmark. And basically can still, so it makes a loop back, and it still orients back towards the next correct landmark. And this is simply because of that error vector calculation. And so here's a bit of a visualization of the way that CyberOctopus uses path integration to run a more complex map. So there's more order sources here. And so we'll see actually the memory vectors drawn out, right? So this is a little bit sped up. But here are those memory vectors between this pair and then this pair. And so it's using, so it can use all these memory vectors to make shortcuts, but also note that I kind of just missed it, but once it encounters that award, it can actually, is essentially using kind of the same mechanisms as before, right? Chain together associations, but now that include the memory vectors, we can actually build a full map of shortcuts, even between, right, the pairs that it hasn't visited before. And so this is just by using simple vector algebra. And we can see as it kind of goes along, it will update these to kind of more accurate locations. And so now all of these memory vectors provide potential shortcuts, right? But what if the environment was a little bit more complex with walls? And let's say, what if there's an unforeseen obstacle blocking a shortcut? If the agent is forced to detour from its memory vector, it can basically easily use its path integration system to essentially split the memory vector into sequences of detour and correction vectors and code them into the FAM. So here's another visualization of this with the CyberOctopus. And to save some time, let's assume that it already has a memory vector learn between these two pairs, between this pair of order sources. So it already knows where to go, essentially. And so it's being guided by that memory vector. But then it encounters a wall. And so it's forced to make a detour. And we can see it actually, okay, here's again. We can see it actually start splitting that memory vector into a detour and a correction vector. And as soon as it orients back enough onto so it corrects its course enough, it actually stabilizes that encoding. So that detour and correction vector are encoded into the FAM, and it can be used for the future for navigation to not make this mistake again. And so this is quite useful for navigating much more complex environments such as mazes. So as we've shown, we basically taken a bottom-up approach, starting with Cybersex and simple associative learning, and expanding it step by step to this affective valuation, and then simple episodic memory and spatial learning, including path integration. And this is following a possible evolutionary path of cognitive map formation. So in particular, we saw that to go from higher order conditioning to sequence memorization, it's necessary to form associations between neutral stimuli as well, along with the learning of the relative order or timing between those stimuli. For more complex processes like cognitive mapping and episodic memory, these involve a greater degree of encoding context between stimuli, such as distance and direction. And so we expect that we can similarly encode additional contexts and chain them together to expand the FAM for more abstract cognitive mapping. So for instance, learning social hierarchies or more abstract cognitive tasks. And so this is the complexity of associations that we see with increasing with the complexity of learning. And so I'd like to thank everyone who has joined me here today. My PI, Professor Rainer Gillette, and his lab, which includes Colin, Gili, as well as the lab alumni, Jeff and Misha. And I would like to thank our collaborators, Gish Chowdhury and the entire Cyberoctopus Miri group, our program directors for the Miri grant that funded our work, our affiliates, and of course, the Center for Artificial Intelligence Innovation for this wonderful opportunity. So thank you all, and please let me know if you have any questions.
Dr. Gribkova, wonderful presentation and already great comments coming in the chat box. So John, thank you for sending that as well. Just as a reminder, this presentation will be on our website, ai.ncsa.illinois.edu under seminars in a few days and recorded fully. So thank you, Dr. Gribkova, for such a great presentation. Looking for questions. If you have any, you can either enter them in the chat or just unmute and ask. We're an informal group, so if you have a question, please ask at this time.
Yeah, could you please comment something about this, software tools that you use to implement this, both on the analysis side and also on this very nice graphical user interface to this?
Oh, so we use NetLogo to actually program. So this NetLogo is kind of it's a bit ancient. It's quite it's quite simple. I think it was developed a while ago, and I think it was developed a lot for like educational purposes. But it's really nice for us because you can basically, if you've seen. For the interface itself, right? It's you can basically just drag and drop buttons like graphs, and then it's quite a nice visual user interface. Yeah. So was that the question? I may have.
That was a question. So this interface indeed very, very nice. But what about the simulation behind it? Is it also in the same language? Or?
Yes. So this is NetLogo. This is terribly simple, terribly simple language, but it's like, so if anyone might know, it uses turtles as like agents. So it's I feel like it may have I forget, may have been a different name for it, but it's so this is all in NetLogo. And essentially it's I would say it's probably one of the most the easiest languages I've seen, and one of the most quite useful, especially with the visual interface.
Yeah, this is very impressive, really, for and it all says that it's not about the tools and all that; it's what you can do with these tools, really.
Yeah, exactly.
So, so do you have any future plans for developing this tool?
Yes. So I guess like the most immediate next step would be to make a much more complex environment. So I do, let's say, a much more maze-like environment. Some of the other next steps. So this is where it gets a bit more expansive. So one next step we do, I think, want to explore is social interactions and social learning. So if we have a group of these little guys, how can they actually interact and actually learn, let's say, to avoid, let's say, some other conspecifics. And the problem is that each one of these would have this kind of like feature association matrix, and then so where calculations get a bit complex. So we might need something like a bit more powerful than my old computer. Other stuff I would say is maybe increasing like the motor capabilities a bit. So if we can make an environment that's a bit more interactive, right? If we can actually push on the blocks or carry the food items, right? So if we can actually show, let's say, so if you've seen caching behavior in ScubJs, which is quite interesting, we might be able to show that a bit. And I think kind of most of the next steps are going to be kind of increasing complexity of the environment to actually give the opportunity of the FAM to kind of learn and develop. Yeah.
And Katya, I want to make sure you see Vladislav's comment in the chat as well.
Oh, yeah. So so the agents, like the default agents, are called turtles. Yeah, for network.
Katya, thank you so much for your presentation today. And just thank you for your time and your expertise and your sharing. This meant a lot to our center, and we appreciate your time very much. So thank you for being here, and thank you all for being here. Have a nice break upcoming, and we will see you into the next semester. So thank you very much for today.
Thank you.